want to thank all the speakers for some really interesting talks. I think in making some comments, and I'm going to keep them brief so there's time to ask the speakers some questions, I want to start by reiterating Tim's two thoughts. It's preaching to the choir to say to this group that individual differences matter to society in all ways we can imagine. So I'll skip that one quickly. But everything, we've known for quite a while now that everything is heritable. And that means that we often really need quantitative genetic studies in order to get to the bottom of how the traits we see all around us get to be the way they are, and in particular how they develop. They're the best ways of, as Robert said, showing how the environment exerts its influence. Some of my favorite things about quantitative genetics are that the field has always shown a great responsiveness to criticism that's been levied at it. Going back to the 70s and 80s when people were first starting to talk about the heritability of behavioral characteristics, all of the assumptions that you have to make in order to do a twin study and the flimsiness of the twin correlations and so on, were constantly being discussed as ways of completely dismissing the results. And behavior geneticists and quantitative geneticists repeatedly rose to the challenge of coming up with ways to try and measure some of those assumptions, try to get around them, try to come up with new ways of challenging those assumptions. And I think that still is a primary characteristic of the field, and we need it because we're facing many challenges that our success has brought upon us. Another thing I really like about the area is the twin studies that have been set up, and now, the journal Twin Research and Behavior, what is it called? Twin Research and Behavior Genetics. Human genetics, yes, knows very well. Every few years they do a survey of the twin studies that are out there, and there are quite a few at this point. And one of the real features of those studies that's really important, especially to individual differences psychologists, is they measure a lot of variables. There are other studies that measure a lot of variables too. The Lothian birth cohort comes readily to my mind, but twin studies, I think, probably have a particular corner on that advantage. Another thing that I really like about the area is the creativity that people repeatedly show in trying to come up with ways of getting around their problems. I think Frank's talk was a really good example of that, where he saw an opportunity in these huge studies that sort of de facto and by the method they collected method of data collection have come up with a bunch of family relationships, but that wasn't ever the intent in the gathering of the data. But if you go scrounging, you can extract those relationships. That's a really good idea, and there are many examples out there of quantitative geneticists showing that kind of creativity. I think there are many purposes of a symposia like this. One of them is, ISSID is a really disparate group. It's made up of psychologists with many different foci of interests within individual differences and many different types of background in training. We have sessions on neuroscience, we have sessions on very specific measures of brain physiology, we have sessions on topics like quantitative genetics and molecular genetics, as well as your standard or more conventional areas of psychology, psychometrics, and particular areas of content and theories within those content. As the field gets bigger and bigger, many people find that a lot of the interesting work in what they had thought was their area is coming out of an area of science that they don't have any training in. And I think quantitative genetics and molecular genetics too, for sure, is one of those areas for many psychologists. So having a session where a lot of what we hear is sort of basic information about the field is really valuable for a lot of people. And in addition, of course, we have every year we have a whole bunch of new students and younger people coming into the field, and it's often really helpful for them too. I think a bunch of what we heard today was exactly that stuff, and that's not meant as a criticism. We also come to conferences to hear the cutting edge, and there's where I think quantitative genetics is really facing a lot of challenges. We still have those underlying assumptions that Frank laid out, and increasingly molecular genetics is teaching us, as I think Robert was stressing and will stress more later this afternoon, that we have many genes. Most of the traits that psychologists are interested in are likely composed of many, I mean thousands, of genes of very small effects. And many traits that we think of as different, the same genes are contributing to them. The way I read that is, if you want to pick your trait, anything, the way two individuals have arrived at the same level of that trait, there are going to be genetic influences on both of the paths by which they arrived, but there could be no genes that they share. They could be completely different genetically in ways that are involved in their particular position on that individual differences variable. That, coming from the way quantitative genetics has been traditionally approached, that's a revelation. That says we're not going to find the common set of genes, ever. It also places, epigenetics is also teaching us that how the environment is involved in genetic expression is tremendously important. And you hear that from one of Robert's old findings at this point, that many so-called environmental characteristics are heritable. That was a revelation at the time, and it still puzzles a lot of people. But it tells us that the genes in the environment are very much intertwined, and gene-environment correlation is a major issue that we're going to have to identify in this field, or we're going to have to be able to get our hands on how that happens, how society is involved in the paths that people have available to them to exert the genes that they have, if you want, express the genes that they have, and hopefully in constructive, socially constructive ways. I think that's a place that we really don't have any good methods of addressing at this point, and we need them really badly. So maybe I'll leave it at that.